Hello, anatomy students. Today we're talking about protection of the brain, so grab your note sheet and a pen or pencil and let's get started. One of the very first things that is going to help to protect the brain are the coverings that surround it. And you have this picture on your note sheet. We have a number of different layers here. We have the bone of the skull, and then deep to that we have the meninges, which are actually three connective tissue layers: the dura mater, the arachnoid mater, and the pia mater. We also have some cerebrospinal fluid, which is going to help to cushion the brain and nourish the cells of the brain tissue. And then we also have something called the blood-brain barrier. So these are the four things that we're going to talk about today. Okay, so we are on slide 4 now. These are the three connective tissue layers around the brain. So first of all, what we're looking at here is a coronal section of the brain, and then we're looking at the part that was kind of cut out. And our most superficial layer is the skull, and so you can see that over off to the side. And then we have the dura mater, which consists of two layers. The first layer is the periosteal layer. So we have a layer of the dura mater that is actually lining the inside of the skull. And then deep to that, we have the meningeal dura mater, or the meningeal layer of the dura mater. Deep to that, we have the arachnoid mater and then the subarachnoid space. Now, the arachnoid mater, it means spider mother, and it's named that because the subarachnoid space has fibers that are attaching the arachnoid mater to the next layer that actually touches the brain tissue itself. And I should probably pause and mention here that dura mater means tough mother, and so the dura mater is the toughest layer that we have of the meninges. And then the last portion that actually touches the brain tissue itself is called the pia mater, which means gentle mother. All right, so this is slide 5 on your note sheet, and a couple of these things I've already mentioned, like the periosteal layer and the meningeal layer. They are going to be fused together except in a few places, and then they're going to form some spaces that are able to be filled with fluid. These spaces are called venous sinuses, and they are going to collect venous blood. And if you look at the picture that we have here, you can see that right just above the longitudinal fissure of the brain, we have what's called the superior sagittal sinus. And so that's going to be an area that's going to collect venous blood and then return it to general circulation in the body. So we have the fissures of the brain, which I had you label in a previous video. So we have the longitudinal fissure, we have the parieto-occipital fissure, and then we also have the lateral fissure as well. And so in these fissures, we don't just have our meninges just kind of gliding over them, just covering them. They're going to actually go down and fold inward and then come back out. And so where they fold inward, they're going to be very thick and very tough, and they form these structures. So that word F-A-L-X is pronounced fox. So the fox cerebri is where the meninges fold inward into the longitudinal fissure. The fox cerebelli is what is going to divide the cerebellum into two parts. And then the tentorium cerebelli is going to be between the cerebellum and the cerebrum. So it's going to separate the cerebellum and the cerebrum. And so when we look at the picture of the cadaver in the lower right corner, you can kind of see that we have these really thickened areas of the meninges where they fold into the fissures of the brain. So I told you that the arachnoid mater means spider mother, and there are some thread-like extensions, like cobwebs, that stretch through the subarachnoid space to the deepest layer of the meninges, the pia mater. Well, the subarachnoid space is filled with CSF, which is short for cerebrospinal fluid. And throughout the subarachnoid space, you're going to have some like outpocketings that are called the arachnoid villi or the arachnoid granulations. And they are going to protrude through the dura mater. And this is where cerebrospinal fluid is going to empty into the venous blood in the dural sinuses so that we have a constant refreshing of cerebrospinal fluid, new cerebrospinal fluid bathing our brain all the time so that we don't have like stagnant fluid kind of sitting around. So those are the arachnoid villi or the arachnoid granulations. And if you look at the picture, I've got an arrow pointing to one of them. See how it extends into that dural sinus? It kind of looks like a little chef's hat or maybe even like the end of a long bone, but they're just these little projections that are going to allow for cerebrospinal fluid to dump into those dural sinuses so that it can become a part of the venous blood supply, and it goes and it gets filtered and stuff as it joins the general circulation of blood. And then the pia mater, this is the gentle mother. It's the only layer to actually touch the brain tissue itself, and it's going to support blood vessels that enter into the brain. Okay, now let's talk a little bit about CSF or cerebrospinal fluid. What you're looking at here is another coronal section of the brain, but like the part that kind of like dips like downward at the bottom, that's supposed to be the spinal cord, but of course it's truncated because it needed to be shortened so that you could see it on the whole picture. But before we move on, I just wanted to call your attention to the fact that the brain has spaces in it. It's not a solid structure, and the spaces are called ventricles. And there are 4 different ventricles in the brain, and I just wanted to make sure that you knew that before we moved on. Okay, so cerebrospinal fluid is very similar to the plasma, to the liquid part of our blood. It's going to have less protein and more vitamin C than our plasma will, so the composition is going to be a little bit different, but it's going to function to cushion the brain. It's going to allow the brain to float so that like gravity doesn't like pull it down towards the base of the skull and like kind of cause it to squish itself under its own weight. It's also going to inform the brain of carbon dioxide and pH levels that we have inside of our bodies in order to trigger homeostatic processes to make sure that those values remain balanced. All right, so these are the ventricles that I was talking about here. And so now we are on slide 19 of your note sheet. And so go ahead and take a look here. This is the anterior view of the brain. And as you can see, we have 2 lateral ventricles. Well, we don't even like say like one of them is ventricle 1 or 2. We just call them the lateral ventricles. The 3rd ventricle is the one that surrounds the yo-yo structure, that is the thalamus. And then we have a connection called the cerebral aqueduct, which connects the 3rd ventricle with the 4th ventricle. And the 4th ventricle is the one that lies behind, or in between, I should say, the cerebellum and the cerebrum. Come on. Within each of the ventricles, we have some knots of capillaries that are called the choroid plexus. And the choroid plexus, along with ependymal cells that are found there, are going to be responsible for secreting and synthesizing the cerebrospinal fluid. And basically, it's a filtrate of the blood. So the ependymal cells are going to secrete molecules into the cerebrospinal fluid. And it's also going to be responsible for ionic transport in and out of the cerebrospinal fluid so that it's not like there's not too much sodium or there's not too much potassium, etc. And then because the ependymal cells are ciliated, the cilia are going to wave and they are going to help to circulate the cerebrospinal fluid through the ventricles and around the outside of the brain as well so that it's always moving so we don't have this stagnant fluid, in this dark, warm place, maybe hiding infection and causing harm to the brain. So we keep it moving, and then the cerebrospinal fluid is going to enter into the subarachnoid space, and then it enters the bloodstream through the arachnoid villi. And so this is the way that the cerebrospinal fluid is going to flow both through the brain and on the outside of the brain. And so, number one, these are the lateral ventricles. Number two is called the interventricular foramen. So there's going to be a little hole or a little tunnel that's going to connect the lateral ventricles on the right and left sides of the brain. Number three is the third ventricle that's going to bathe the thalamus in cerebrospinal fluid. Number four, the cerebral aqueduct. Number five is the fourth ventricle, but in order to see it in this drawing, they didn't draw the cerebellum because we're looking at the back of the brain now. Number six is the central canal of the spinal cord. So the spinal cord has a tiny hole that runs down the whole length of it, and it too is filled with cerebrospinal fluid. After the fourth ventricle, it could either go into the central canal or it could exit to the outside of the brain through number seven. Number seven is called the lateral apertures. Or it can kind of bathe the dorsal side of the spinal cord by exiting through a small hole, which is labeled number 8. That's the median aperture. And if it goes out the medium aperture or the lateral apertures, the cerebral spinal fluid is going to enter into the subarachnoid space. And eventually, it will reach an arachnoid villi, or villus, singular, and that cerebral spinal fluid will then enter into the venous blood, and it will leave the brain and enter the general bloodstream where it gets cleaned out and it just kind of becomes a part of the blood itself. And so this is a mid-sagittal section of the brain. On your sheet, we are looking at slide number 22. And so once again, number 1 is a lateral ventricle. Number 2 is the interventricular foramen. Number 3, we are looking at the third ventricle. Number 4 is the cerebral aqueduct. Number 5, we have the fourth ventricle. Number 6, we're looking at the lateral aperture. Number 7 is the medial aperture. So you can see how number 7 kind of allows that fluid to move out and like bathe like the back or the back outside of the spinal cord. Number 8 is the central canal. Number 9 is the subarachnoid space, and then number 10 is the subarachnoid villus, and then number 11 is the venous aperture, or the venous sinus, which is going to bring the blood back to regular circulation. All right, so this is just another diagram that's emphasizing that our cerebrospinal fluid, after it enters the subarachnoid space, will enter into an arachnoid villus and then into the venous blood supply. All right, I'm going to stop it here and come back and talk about the blood-brain barrier later.